So on to chapter 23. We just learned all about Darwin and all about all of his ideas and where they came from and everything like that. But now we're going to get into how natural selection actually works, how evolution actually happens and the effects that it has on a population. So when we talk about, like here, this is a picture at the beginning of the PowerPoint. Why did this happen? Why did humans evolve from their common ancestor with apes? This exactly didn't happen, and we'll talk about that, but how does one generation of populations, the generation or the population of humans, how does that change over time? That's what this chapter is about. So there's this picture at the beginning of your chapter, and it says, did this bird evolve in its lifetime? So was there evolution actually happening to this bird in its lifetime? And the answer is no. What happens is evolution acts on the population as a whole, and whichever organisms in that population pass on their genes to the next generation, that's when evolution happens. So the genes that are passed on are the ones that are going to form the next several generations. So that's what we're talking about. So it's called microevolution. So this is evolution on the smallest scale, micro meaning tiny. And this is when we look at the change in allele frequencies in a population over several generations. So we're looking at the frequency of a dominant allele or a frequency of a recessive allele in a population over many generations and see how that changes. And there's three mechanisms that cause allele frequency change. These are natural selection, so obviously we're going to talk about that one, genetic drift, and gene flow. And only natural selection causes adaptive evolution. So genetic drift and gene flow can drastically change the allele frequencies in a population, but only natural selection will necessarily make them more adapted to their environment. And we'll see what that means. So the first section is called genetic variation makes evolution possible. So remember that that's one of Darwin's principles, is that there's variation in a population, and that's really, really important. Because if there's no variation, then there's never going to be any change, right? So we're changing based on what's already in the population, and so if there's no differences, every single generation is going to be exactly the same. And so right about the time, or right after Darwin published on the origin of species, Mendel came out with his ideas on genetics. And so then we were able to understand how this evolution actually happens. So you can actually inherit a gene from your parents, and that's how evolution is going on generation to generation. So it's important to remember that evolution only works on heritable characteristics. So there's a lot of characteristics about living things that are not inherited from the previous generation. For example, this is the Nemoria moth, and the reason that they look so different, obviously this one looks a lot different than this one right here. And the reason for that is because of a chemical difference in their diet. So whatever they're eating, so this guy's living on a tree branch, this guy's living on a leaf, whatever they're eating is making them look different. Probably, maybe so they can blend in with the tree. I don't really know. But that is not heritable. So if this guy has offspring and they start living over here, they're going to look like this guy. So the way that they look, their physical appearance is not being passed on. It's totally environmental. So we know that there is always variation within a population. We've already said that. And we can measure these in a couple of ways. Sometimes there's discrete characteristics, which is either like you have it or you don't. You're red or you're blue, you're tall or you're short, etc., etc. When Mendel studied his pea plants, he purposefully chose discrete characteristics. So he purposely chose red or white flowers. He purposely chose round or wrinkled seeds, green or yellow seeds, because there's only two, right? There's only two phenotypes. But then sometimes we have something called quantitative characters, which vary along a continuum within a population. What that means is humans are not all tall or short, right? We have some very, very short humans and we have some very, very tall humans. And the reason for that is because it's a quantitative character. So basically, where this comes from is that quantitative characters usually have two or more genes that affect the phenotype. So the reason why you're tall or short is because of many different phenotypes, many different genes inside your body. It's not just based on one. It's not dominant is tall, recessive is short. There's a ton of different genes that affect it. So that would be an example of a quantitative character. So sometimes we need to measure the differences or the genetic variability in a population. So a population, I didn't define this yet. A population is a group of one species living in one space and interacting with each other. So it's important that they're interacting with each other. That way there can be gene flow, meaning they can pass genes to each other through reproduction. So that's what a population is. So sometimes we might need to measure the variation within a population, and we can measure this in a couple different ways. We can measure it at the whole gene level, so what different kinds of genes are present, or we can measure it at the DNA level, so actually how many nucleotides are the same between individuals or in between a population. And we can measure this in one way called the average heterozygosity. So this is how, picking one gene, one locus, how many individuals or what percentage of individuals of that population are heterozygous at that locus. And that gives you a general idea of what the variability is, because heterozygotes have both dominant and recessive alleles. Okay, so variation between populations. So we just talked about variation within a population, but there's usually always also variation in between populations. So this means differences between gene pools of separate populations. So a gene pool are all of the genes or alleles present within one population. So the gene pool of ants within room 100 may be very different between the gene pool of the ants in room 400 because they're so far apart and they're not interacting very much. Those are two different populations. For example, there is a picture from your textbook, and these are two different populations of mice. They are both the house mice on the Atlantic island of Madeira, and they were brought by Portuguese settlers, and they came with them. And so, but they were geographically isolated. So there's a big mountain range in the middle of here. So there's the mice on this side of the island and the mice on this side of the island, and they are very different from each other because they have evolved a little bit over time. Their gene pools have become different, so there's variation between the populations. You can see that they've had a lot of chromosomal changes, a lot of chromosomal fusions. So this right here is chromosome 15 fused with chromosome 18, and only the red species has it. Up here, chromosome 5 is fused with chromosome 18. So you can see there's difference between the two populations, even though they live close to each other. And then there's also something called a cline, which is an example of geographic variation. And this is when over a spectrum of differences that there exists variation. So for example, the mumachug fish vary depending on what temperature they live at. So it's very cold in Maine. The water is about 6 degrees Celsius, whereas in Georgia, the water is about 21 degrees Celsius. So this is 40-ish degrees, and this is about 60-ish degrees. I might not be very good at that. But that's what it is, a huge temperature difference. And you can see that the fish have a much higher frequency of the LDHB, lactate dehydrogenase B, little b allele, up here at the very cold temperatures, as opposed to down here, that allele is much, much different. So you can see that over a spectrum, the allelic frequencies change. That's called a cline. So the sources for new genetic variation. So how do we get genetic variation? Where does this come from? So in your textbook, there's a lot more reasons than on here. The first one is formation of new alleles. So new alleles can be made. This happens through a mutation. And like it says, the average is about one mutation in every 100,000 genes per generation. So it's very small. And actually, the mutations have to happen in a cell that's going to be passed on to the next generation. So if I have a mutation in a cell in my hand, that sucks for me, might cause cancer in my hand or something, make an eye or an ear grow on my hand, probably not. But the cells in my hand are not going to be passed on to my offspring. However, if there's a mutation in a cell that will be passed on to the offspring, gametes, or the cells that will become gametes, then that's going to be passed on because those are the actual physical cells that are going to the offspring. So those are the mutations that are the most influential because they're being passed on to the next generation. And then, sometimes mutations can be very harmful. Sometimes they can cause things like sickle cell. Sometimes they can destroy whole proteins. They can be frame shift mutations or point mutations, all that stuff we learned about. But sometimes they can have little to no effect. Again, if they're in a cell that isn't going to be passed on to the next generation, if they're silent mutations, if they're in introns that don't code for anything, et cetera, et cetera. Or if they just code for a similar amino acid, it doesn't really matter. Then something else that can cause or a source of genetic variation is altering the gene number or position. So if for whatever reason we amplify a gene, that means that now you have more copies of the gene, then you're going to have more of that gene product and more of that protein or whatever it codes for. And so that can cause a big difference. For example, the ancestors of mammals that lived however many years ago, so the common ancestor for all of the mammals that are alive, had one gene for detecting odors. And for whatever reason, this gene has been duplicated a lot of times in different populations or in different species, and now humans have 350 genes for smelling things. Mice have 1,300 genes for smelling things. So this has been duplicated and is a difference in phenotype. For example, we can probably smell a lot better than the ancestor that only had a single gene for it. And mice can probably smell a whole lot better than we can because, my textbook says 350, this says 1,000. But they have a lot more than that. And a lot of our genes have since been turned off. They're not being transcribed, about 60% of our smelling genes, whereas mice, only about 20% of them have been turned off. So it seems that it's very much more important, or it's much more evolutionarily advantage for mice to be able to smell things. So the amplification of that smelling gene causes a change in phenotype. Mice can smell better than we can. That's a phenotype right there. Sometimes rapid reproduction can cause changes in new alleles. So, for example, humans and plants and things like that, we have very low mutation rate, about 1 in every 10,000 genes, but it's even lower in prokaryotes. However, mutations tend to accumulate more in prokaryotes because they reproduce, they replicate so much more quickly than we do. Their generation time is so much shorter than us. For example, it could be a couple hours. The replication time or the generation time of HIV is 2 days. So every 2 days they're replicating. It's very different than humans, which is about every 30-ish years, right? So if there's a mutation once a generation, it's going to happen once every 30 years in humans, and it's going to happen once every 2 days in plants or in HIV. So that's 180 times a year times 30. Think of how many more mutations there are going to be if your generation time is that much shorter. So that causes a lot of new alleles also. And lastly, sexual reproduction can bring about new alleles because of things like crossing over, because of things like genetic recombination, because of like bringing two new alleles together. We can get new formations and new combinations of alleles that we didn't have before that could bring about new phenotypes. So it's just also important to remember that when we talk about all of these mutations, most of the time, mutations are not going to be a positive thing. Because if you think about a living thing and all of the adaptations it has and how well it has evolved to its environment, that's been the consequence of thousands of generations of adaptation. So humans, or I'm not going to use humans, mice that live in Southern California have spent thousands of generations evolving, adapting to their environment, the most fit surviving to pass on their genes. So if there's a mutation, the likelihood that that mutation is going to be better than what has come up over the thousands of generations is very low. So there are sometimes mutations that are very positive. A new allele arises with a new phenotype, and it's very positive for the species and its environment. That does happen. New novel traits arise all the time, but it's just very unlikely. Mutations tend to be deleterious as opposed to positive. So that is that section.